Tonight's talk is by one of our own, by Barbara Hahn. Barbara has been here 3.5 years plus a couple weeks. I know that because she preceded me by about a month to the Cary Institute. Barbara came to the Cary Institute from a postdoc at the University of Georgia. She did her PhD at Oregon State and did her undergraduate work at Pepperdine. Her work is really interesting. You know, yesterday evening, my daughter borrowed my phone and was watching something on YouTube, and up popped an advertisement. And that advertisement was for the SeaWeb Sustainable Seafood Conference this June in Barcelona. And it wasn't it wasn't inappropriate, but it wasn't something my daughter would be interested in. But I'm on the board of something called the Ocean Foundation that sponsors SeaWeb and the Seafood Conference. And I Google quite a lot and read quite a lot about ocean conservation on my phone. And I thought it was a sort of harbinger of what was coming tonight. Barbara Hahn uses those kinds of tools that allows Google to send, or YouTube, which is Google, Alphabet, sorry, to send me things that I would like to see, and often they get it right. Barbara uses this in a much more, I think, socially responsible and altruistic kind of way to look at how we can predict where disease is going to spill over. Because as my mom always used to say, and I'm sure your mom always used to say, an ounce of prevention is worth a pound of cure. And knowing where these things are going to spill over gives us a shot at preventing the spillover before it happens. That's all I'm going to say, other than the fact that Barbara is a distinguished and authoritative speaker. She is an advisor to the Office of Science and Technology Policies, Pandemic Prediction and Forecasting Science Working Group. That's at the White House, both the last one and this one. It appears that epidemic disease is something of interest to everyone. She has advised the Department of Defense. There's something called PREEMPT, which again, Preventing Emerging Pathogenic Threats, which DARPA, which is the Defense Advanced Research Program, agency, right? You have a project agency. DARPA, the people who actually did bring you the internet, likes to fund things that are on the edge, are edgy and forward-looking, and it is not at all surprising that Barbara has been talking with them and advising them on how to put a program in place to look at how we can predict emerging infectious disease. So it is a great pleasure to hand the stage over to Barbara, and please welcome her to her home, the Cary Institute.
Am I on? Yes. Thanks for that really nice introduction, Josh. I hope that I can do that authoritative speaker part some justice here. I want to start out tonight by kind of getting a vote from people here in the audience. I want to ask people, how many of you think that compared to the last 20 years, our disease threat today is about the same, a bigger threat than it was 20 years ago, or less of a threat? So how many people think that it's about the same? Okay. How about less? Less than 20 years ago. Like, we're doing better now than we were 20 years ago. People are, like, indecisive here. What about more of a threat? Oh, good. This is a good audience. Okay. Yes. So you guys are doing better than the general public. So we worry about it, right, as Americans, and really we should as a global society. But 51%, only 51% of people think that it's more of a threat today than it was 20 years ago. And that's, I don't know if I find that comforting or troubling, because actually the reality is that we're dealing with an issue that is not going to go away, and it's just getting worse. So this is just an updated map that was published recently by the head of the National Institute for Allergens and Infectious Diseases, which is one of the institutes that comprises the National Institutes of Health in Bethesda. And they're just basically plastering on this map these diseases that are either considered to be emerging, which means that they're expanding in range geographically, or they are causing more frequent outbreaks in the same populations in which they've been observed in the past, so that's emerging, or re-emerging, which means that they died down, they died back a little bit, but now they're becoming a problem again. So this is just a smattering of diseases that at least the National Institutes of Health are concerned about. But of course, we can ask some skeptical questions, right, as a scientifically interested audience. Is this map so busy because we are looking harder for diseases? Is it because we have better technology to detect things that we think are making us sick, and so we run fancier tests and we can pick more viruses up? There are a lot of reasons that bias and technology and maybe even things like the internet can help us be more vigilant about these health threats in the future. But actually, a group of scientists at Brown University recently really dug into this question of, are diseases really on the rise globally? I mean, are we really under the threat that we feel like we are? Or is this just a byproduct of us being better informed as a society? And so they did this analysis where they scraped together data that describes outbreaks of all known human infectious diseases. And this data set is probably like 20,000 records long. They started from the 1980s and they went forward in time. This is by Kate Brown and colleagues at Kate Smith and colleagues at Brown University. And so they produced this map that shows the cumulative number of outbreaks for human infectious diseases since 1980, and this is just the number of unique outbreaks, okay? So darker countries have more outbreaks, and you can sort of see a general pattern that the Northern Hemisphere might be better studied. So this obviously raises the question of like, well, it's the richer countries, like we're more interested in keeping our health and we have more money, and so that's why we're darker. Well, actually, they dug in a little deeper and they said, okay, well, if we control for study effort, if we control for gross domestic product, if we control for information flow and information accessibility, if we even control for the advent of the internet and connectivity across all of these countries, do we still see an increase? And the answer was overwhelmingly, yes, we do. So the jury is not out anymore on this question. We are definitely seeing an increase in infectious diseases globally, not just the human infectious diseases, but ones that are arising from animal sources. Those are the kinds of diseases that I'm most interested in, and I'll tell you a little bit more about them later. But I want to dig into this question a little bit of why do we think these diseases are becoming more of a problem than they were before? Can anybody give me some guesses as to why we're having more disease issues now than before, even with all of our technology and global travel? Oh, that was a good one. I have a slide for that. It wasn't in order, though. You have to save it. You guys are listing off a bunch of the things that are on this slide, right? So international travel is a big one, right? Human susceptibility, we're not as healthy, I guess, as we were before. Even though our lifespans are longer, we are also getting sicker the longer we live. Land use change is a big one. So I don't have to tell this audience that deforestation and even the ways that we use land that is already deforested has a big impact on our susceptibility and our contact with pathogens. There's a whole bunch of reasons, right? Antimicrobial resistance and bacteria is a huge problem, as Emma Rosie is an expert and can tell you. Things that some of the chemicals that get into our water have a big role to play in that aspect. So the global travel thing, I want to make this point here that public health is global health. Right, so we are an incredibly interconnected society. This is just showing us global trade routes, okay? So this is like basically all of the travel and supply chains and everything, all of the data that we have that describe how things move across the world. That's what these lines are. And the darker areas represent areas where there are fewer people, and the brighter areas are where there are more people. And I want to draw your attention to this statistic here where we know that now 90% of the Earth's surface is defined as accessible, okay? So, and only 10% is considered remote, which is defined to be a location that is more than 48 hours away from a major city. And so if we just look at this map for a second, it's not hard to make the connection that if one person is sick or if a group of mosquitoes gets onto a shipping barge or something, it doesn't take long before it gets to a major city port. I'm really interested in zoonotic diseases, and I want to take a second and just define what this term means because I'm going to use it a lot, and I'm probably going to use some other jargon too, but it's not too tedious, so let's just go through this really quick. So this is sort of a diagram of a basic disease transmission cycle, okay? So you have some disease that's circulating among species in the wild. Here it happens to be bats. And if the disease is sort of living happily in the wild and subsisting in perpetuity in these organisms, those organisms are called reservoirs, right? So they're the reservoir of the pathogen that we're interested in. When that pathogen spills over, it can spill over to infect domesticated species, wild species. This is an antelope, this is a primate. These animals can sometimes get sick, sometimes they're not sick, sometimes they just serve as little bridges. But they're animals that we can become, we're in closer contact with in some cases. So when this spillover event happens, in the vast majority of cases, that's where it stops. That one person gets sick, and it's a dead end. That's, I'd say 99% of most spillover events happen that way. But some of them lead to ongoing transmission, which means that a human can get it, and that pathogen then is able to transmit from a human to another human. And those are the ones that go on to cause epidemics and pandemics. Those are the ones that we're worried about that will cause sort of these global big pandemics, like avian flu. If it acquires this ability to transmit from human to human, a pandemic is it's imminent, I would say, if avian flu acquires that ability. So can anyone give me a guess about what this transmission cycle is describing? What disease? That's a good guess. Malaria is another good guess. Yeah. Did you say? Ebola. Yeah, that's right. And that's not obvious, right? Because we've been hearing a lot about Ebola recently. And I want to draw your attention to all of these remaining question marks here. I mean, Ebola is something that we should not be caught by surprise by again, right? We've had it since the 70s. It caused a huge outbreak in West Africa recently. We even had some of our healthcare workers get infected. And we still know very little about its transmission cycle. We don't know a lot about its basic biology. We don't know what these bridge reservoirs are. We have some evidence to suggest that, these things get exposed to filoviruses, of which Ebola is one. We know that a lot of primates can crash out and die. Whole populations can get wiped out from becoming infected by Ebola virus. Actually, primates have a higher mortality rate than humans do, over 90%. And so if we, even if we were to get a handle on poaching and our biodiversity issues for grade 8 populations in the wild, if we don't get control of this Ebola issue, they're still going to go extinct. So when they get into humans, the problem when it gets into humans is that Ebola burns really hot, to put it lightly. It can infect a couple of other people in one go, and then because of the social customs of the area where the last outbreak happened, a lot of people can get infected from one person dying in the community. Now, this is really interesting. There's a record in the literature that suggests that one person fell ill with Marburg virus, which is closely related to Ebola virus. It's in the filovirus family, after getting bitten by some unknown insect. And so scientists have still not ruled this out, that it could be vector-borne, that it could be subsisting in an insect population somewhere. But we just have so many unanswered questions about this whole landscape. So this is the way we normally deal with, this is just a lot going on here. Let me break this down for you. We conceptualize the disease spillover, the riskscape for infectious diseases. A colleague and I John Drake and I did this maybe 2 years ago. And we were really trying to do this thought exercise of, if we're really interested in preventing infectious diseases, what's a good way to think about that whole landscape? I mean, there's lots of different kinds of diseases, and there's lots of different ways to predict whether these things will happen. What kinds of data would you need to be able to do that well? Well, it really depends on where you're talking about, where you are on this landscape, right? So Ebola, I just told you about, would be in this emergency zone. In the emergency zone, we are not worried about finding out about its basic life cycle. We don't even care what the vector is. We just want to damage control. We want to put out that fire as fast as possible. If an ounce of prevention is worth a pound of cure, we literally dump tons and tons and tons of money into putting out these fires once they get out of control. So the World Health Organization categorized Ebola and later Zika virus as a public health emergency of international concern. And what that means is that this disease is getting so out of control that we are almost unable to contain it. So it's an international concern now, and we all have to rally to put out this fire immediately. And so during the Ebola outbreak, a bunch of us responded by trying to do analyses to figure out where it came from and how we can stop it from happening in the future. But at that point, all of the resources, and rightly so, were directed towards trying to minimize human losses, really. So when we're in this emergency zone, we rely on data that comes from humans. Okay, we want to know who's getting infected, where they are, how fast it's spreading, where we need to deploy logistics so that we can get this thing under control. But the majority of our research and our interest, I guess as a research community, as a medical community, happens in this warning phase when we're not in an emergency setting. So during peacetime is what they call it. During the peace times, we're in this warning phase, and here we're interested in collecting basic information about, what causes a disease to break out with regularity? Why do we keep seeing yellow fever when there's a vaccine with 100% efficacy that we are administering for years to these populations, and still we have people dying of yellow fever every year? What are the reservoirs, and what makes those reservoirs shed virus to the point where it spills over into humans? I mean, these are basic questions that disease ecologists and public health practitioners and doctors are interested in answering. But within this group, there's still a lot of discrepancy in who gets the research dollars and who doesn't, right? So if you are a rich country and you're really interested in things that infect you, like flu, flu is also a good research target for other reasons, the pandemic potential, but you dump tons and tons of money into the diseases that you are most concerned about that are on your borders. But the things that get neglected are actually the things that infect the majority of people on the planet, and those are categorized as neglected diseases or neglected tropical diseases because often they impact tropical nations. So in this zone here, this is where the new stuff comes from, right? So we know that the viral diversity on the planet is almost incalculable. We have a hard time even putting an order of magnitude number to the number of viruses that we expect to be in existence at this point on the planet. Everything is infected by multiple things, so there's nothing on the planet that is not infected by something. Usually co-infection is the rule, where there's multiple organisms sort of vying for parasitic rights to different pieces of you at any given moment. And so the idea that things spill over and become realized as diseases that we care about, that risk really comes from this zone, where we know that there's a huge biodiversity out there. We know that lots of those animals are being infected by multiple things. And we know that our treatment of the landscape and our interconnectedness are really making us more vulnerable than ever before for new things spilling over. And so some examples in this category would be things like MERS, the Middle Eastern Respiratory Syndrome, that's actually found in most camels. And what's another one? I guess chikungunya is relatively new. Is Shannon in the room? Yeah, it's relatively new, right? So there's new things being discovered in this region, maybe a little bit more, I guess SARS is another one, coronaviruses, a little bit more commonly than we'd like to see. But here I draw your attention to the kinds of data that we use here really are equal parts, what do we find spilling over into humans? Even if it's not making you sick, if you can find its genetic signature in your blood, it's telling you that it's getting into your body somehow. What are the organisms? What's their basic life history? Like, where are these things? What environments are they found in? And what societal conditions are we talking about here? So I talked to you about two examples, which I will go into greater detail with later. This is, Ebola. And does anyone know what this one is? No? No. Yeah, Zika virus. So in this warning phase, I skipped over the examples in my excitement to tell you about the figure. But does anybody know why I put this one up here? Does anyone know what organism that is? Did somebody say flea? Flea. Yeah, it's a flea. What do fleas carry? Bubonic plague, yes. And do you know how fleas transmit plague? If anybody knows this, I will be really impressed. Okay, so bubonic plague is a bacteria, and it makes the fleas constipated. Yeah, so it constipates them, and it also kind of collects in this region of basically their throat. And so when a flea is trying to feed, it feeds more. It tries to bite more often because it's like not able to either swallow the blood and get a full blood meal, so it makes you bite more often, or you're constipated, so you're trying to get it out. So either you're gonna pass it out one end or the other. So you're biting your host, and the blood meal comes in, mixes in with the built-up bacterial plaque here, and then spills back out. So that's how they transmit plague. This is chikungunya virus. Does anyone know what this is? I know, I'm sorry about all the gross pictures. I mean, you guys are expecting this, right? So I was telling you that some of these diseases are neglected, and we don't often think that these neglected diseases happen in rich countries. But actually here in the United States, we have a huge population of people that are underserved and living in poverty. And a lot of those populations are sort of in the south, sort of in the Appalachian region. And this is a roundworm that comes from cats and dogs. It's called Toxicara. And it's actually a pretty big problem in these southern states. And the worm burdens can be extraordinarily high. And sometimes the infections are asymptomatic, meaning you can't tell that you have them. They don't really cause you any problems. Obviously, this person's having a little bit of a problem, but I don't know that this is I don't think that this is affecting their vision. I'm not really sure. But it's actually really simple to prevent that spillover event. I mean, some public health education and also just deworming medication. That's all it takes. So some of these problems are imminently fixable. And they don't require, lots of technology, but that here we are. We have this whole landscape of issues. So the problem with this, with us being able to predict new things that spill over from here to here, and knowing everything we want to know about the dynamics of this region, so that we can prevent this region, the limiting factor for all of this is data. And that's kind of incredible to me, considering the fact that we're able to pretty much predict the weather with not too bad accuracy. I mean, I'm sure that people would have liked to know which exact houses went without power in the last storm. That would have been really nice. No amount of prediction is going to be able to get you there. If they could, then insurance companies would be making a lot of money. But actually, that kind of prediction is just not possible because of the underlying stochasticity. We call it stochasticity. It's basically randomness. There's an element of randomness underlying every event that happens on the planet, and you just can't predict with that much precision. But what we can do, and what actuaries and insurance companies make lots of money off of, is we can draw risk boundaries around where we think that risk is highest and lowest, and where we think it's most probable and less probable. And that's why I think machine learning is a great tool to use. So, machine learning. Machine learning is born from artificial intelligence research. It's an area that's under hot development by computer scientists and used in industry everywhere. And so it's just basically pattern recognition. It's like algorithms to detect patterns in data. You can do fancy prediction with them. You can predict what Josh Googles on his phone. And you can do that by using high-dimensional data, which basically is a fancy way of saying you collect tons of data of tons of different kinds. That makes it high-dimensional. You put it all into an analysis and black box it a little bit, do some hocus pocus, and you get a prediction. That's what most people think machine learning is. It's actually much more prescriptive than that. And companies are capitalizing on this left and right. So even if you don't know how to do machine learning, you interact with it on a daily basis. So machine learning suggests shows that you might like to see on Netflix. It learns the patterns of the kinds of music that you prefer on Pandora and suggests new artists that are maybe less popular but you probably would like. And it does a scary good job of predicting what else I might want in my shopping cart when I'm shopping on Amazon. So machine learning is very successful in lots of settings, and it hasn't yet been applied to disease ecology. And so when I first learned about machine learning, I thought, oh, surely somebody's looked at this. And then for infectious diseases, it's such low-hanging fruit, right? But I did some digging, and it turns out nobody had really connected the two things together. And so when I talked to computer scientists, they're like, oh, yeah, that should be easy to do. That's not a big deal. But when we first started doing this work and we talked to ecologists or disease ecologists or virologists, they would go, oh, you can't do that. That's not something you can do. Like, I don't get that method. It's such a different way of thinking about data that it's actually it was quite hard to get this first piece of proof of concept out there in the literature. Because what kinds of questions can we answer with it? It's a little bit exciting to think about, right? Like, you can ask questions like, what organisms are most likely to give rise to zoonotic diseases in humans? Can you train, if you give the algorithm information about organisms, can the algorithm start to recognize the traits of something that should be able to infect you? Right? So we can apply that type of thinking to hosts, like what hosts should be able to carry zoonoses. You can apply it to pathogens. What kinds of pathogens have the potential to be able to spill over and cross that species barrier into humans? You can apply it to vectors. We have tons of mosquitoes and tons of ticks out there, and many of them are very understudied. Can you come up with a profile for what a good tick vector looks like? If I want to know what vectors are out there, defense is really interested in this. When you send troops out into the middle of nowhere, shouldn't you kind of know what vectors are there and what they might be carrying? And it's just not feasible for us to cast a net over the entire globe and expect to get lucky and survey where we need to survey to get a handle on this landscape. And so what we did was we collected data on organisms. I'm going to give you a couple of examples, but just to crystallize what kinds of data I'm talking about, here are some data that describe things like size, like reproduction, life history, or like the way that these animals live their lives, their biogeography, sort of where they're distributed, what kinds of temperatures they like, and phylogeny, so some indication of their evolutionary relatedness to each other. And you give the algorithm all of these intrinsic features that distinguish one animal from another, right? One pathogen from another. What are the things that make these things different from each other? And then you ask it this basic question of, can you tell me which ones carry diseases and which ones shouldn't, based on their profile, based on these traits? Because what these traits also represent is some long evolutionary history, this long evolutionary history that these organisms have not only with their environment, but with other selection pressures, for example, parasites and pathogens. Right? So these features are capturing this information that recapitulates this long evolutionary history that these organisms have dealt with over time. So we applied this thinking to the Ebola virus outbreak in West Africa. So this was a map that was produced by the World Health Organization, and I think it's really compelling. It's kind of scary, so don't let the red. It's over now. So, but the problem with this disease is that it goes away and then it comes back, and it goes away and it comes back, and it doesn't always cause an outbreak this size, but outbreaks are pretty regular. So we're going to have another one, and this one, because of where it happened, it caused a huge amount of death. So over 11,000 people died. And what the statistics don't report is how many kids were orphaned as a result of their parents dying of Ebola. And so this crisis really, it was not just a one-time thing. It really wrecked the country for many generations to come. I mean, they're sort of picking up the pieces financially and socially from this outbreak. And so what we wanted to do was figure out, well, if we want to prevent the next one, we got to figure out where it's hiding out when it's not in an emergency status, right? So I want to draw your attention to how big of an area we're talking about here. So this is sort of sub-Saharan Africa here. This is the United States, size of China, India. It's a really big amount of land. This is where the last outbreak happened, in this little corner. This is where outbreaks regularly happen. So spillover events are happening kind of all the time in this region. And the distance between these two things is something like 3,000 kilometers. Why haven't we found the reservoir yet? Not just because of the area, but it's because the terrain sometimes can look like this. I mean, this isn't like Disney where you walk up and you're like, oh, I want to find the animal, and like these birds come tweeting down, and like frogs jump out, and like, take my tissue. You know, it's not like that. It's actually really hard to do fieldwork in these regions. I mean, here you're kind of lucky because you have a stream, so you have something that's cutting through the vegetation, but it's not easy to go out into this region that's the size of an entire continent and throw a net over it and try and figure out what animal could be carrying this virus. You have to get the timing right because there's seasonality in these locations, right? So animals are coming in, feeding on things, leaving when the flowering event is over. There's incredibly high biodiversity. Okay, so just to paint a, oh, there's an animation here. Yeah, so just to paint a picture, this is just in the Congo Basin, okay, so one little area where outbreaks happen on a regular basis, little area. But it's one of the most biodiverse places in the whole world. It's just sort of a smattering of things that, numbers that I put together just to describe this region. And we have been sampling, looking for Ebola virus reservoirs in this region for a really long time. And just to give you a sense for how many times we've sampled over almost 100,000 animals, 30,000 to 100,000 animals, estimating. And because the emphasis has been, the suspicion has been always on bats, Over 8,000 bats have been sampled, sometimes destructively. The majority of them have to be sampled destructively to get the right kind of tissue. And we've never found live virus in the wild after all of this searching, okay? So this is not an easy problem to deal with. We need some help. So we started out by let's just figure out which bats. Okay, let's just start with bats. We made this decision. The best data come from bats. Everybody thinks it's bats. Let's just start with the bats. So we collected data on the bats. There are 21 species of bats that have ever tested positive for filoviruses. Okay, and when I mean positive, it's like there's some indication from the serum of the tissue that it's been exposed to a filovirus. We don't know what kind it's been exposed to. We don't know if it was actively infected. We don't know if it was replicating in the body, but they've been exposed. So let's just take those species. And when you map those out, the majority of the samples obviously come from Africa. So when we zoom in on this region, the black dots represent known outbreaks. The blue areas represent, the blue areas with the halo represent multiple outbreaks. And this star is where the last outbreak started. So if you look at this map, the fact that we didn't have a bunch of dots over here from previous spillover events still doesn't get us off the hook for being surprised that it broke out here. Because what you're seeing here in the colored regions are all of the bats' geographic ranges overlaid on top of each other. So these are the 21 bat species that carry Ebola virus that have tested positive for it in lab tests. And the majority of them, a lot of them are overlapping in range right here. So there's a lot of carriers right here, a lot of carriers here, some carriers here, but really this whole region is covered in yellow, right? So I don't know if we should have been surprised about that last outbreak. I'm surprised that it took off so fast, but actually maybe not, because there's so many people living in that region of Liberia. So when we trained an algorithm to try and, we wanted the algorithm to tell us the profile of something that carries Ebola. Not just that, but like we wanted the algorithm to learn the traits of something that can test positive for filovirus, and then identify the other bats that look like the bats that carry filovirus, so that we can figure out which ones look like those carriers, and so maybe these guys should be the targets for future surveillance, right? So that was a classification task. It performed this task really easily, which I thought was shocking. It was 87% accurate in distinguishing the carriers from the non-carriers. So and we tried to break it a bunch of different ways, and we were testing for bias and all these kinds of things. It's a pretty robust result. And I'm just showing this map here of the 90 species that were in that 90th percentile probability of being likely to carry filoviruses. Okay, so we see the star here, and we see pretty quickly that Africa might have been sampled out. We've been doing a lot of sampling in Africa. Some of the predictions that our algorithm made were this guy, the Caraphon pumilus. And I was excited to see this one pop up at the top of the probability distribution because this guy named Swanepoel collected a bunch of plants, animals, and insects, just basically whatever they could get their hands on in this region, and brought it back into the lab and just started hammering everything with Ebola virus. So Ebola Zaire, just infect everything. If anything can replicate it, that's a lead. I mean, he was really grasping at straws. So he infected a bunch of things, and this bat not only was infected by Ebola Zaire, but it replicated virus, and they were able to isolate the virus back out of the animal. So definitely has the capacity to serve as a reservoir, but it's never been found positive in the wild, which is why it didn't make it into our data set. The algorithm also predicted this species, Budocophori. It's closely related to this animal here, the Epimops franchetti. These guys are essentially the same. When you talk to a bat biologist and you show them these results, they get super excited because they say, oh gosh, forehead slap. That's basically this bat, but just in a different area. Of course that would come up at the top of the list. Why didn't we think about sampling that before? So the algorithm is giving us some intuition, or some results that make intuitive sense. The other place that surprised us on this map was obviously Southeast Asia. So when you zoom in on Southeast Asia, here's what you see. The distribution of bats that our algorithm predicts to be positive for filoviruses is really hot here. So up to 26 different species are overlapping in range in this region. And so that begs this big question is, why aren't there more filovirus outbreaks in Southeast Asia? We've got high biodiversity, we've got lots of people, We have primates. We have lots of livestock. Sort of all of the stars seem to be aligned. What's up with Southeast Asia? And especially when we consider the fact that novel filoviruses are being isolated here with some consistency. People are looking for filoviruses in this region because they suspect that it should be here. And so he et al. Reported a new one in the Yunnan province. Yang et al. Reported a new filovirus in a new reservoir species in the wild in this region. And that reservoir that they found positive for the first time was number 5 on our list of predicted species. So this was an independent field study where they went out and were looking for filoviruses, testing animals that they could get their hands on, and it matched up with our predictions. So it's giving us some indication that the model is working. So I'm going to end with another case study on Zika virus. So kind of as soon as the Ebola virus stuff died down, Zika virus started to make the headlines. And of course, the World Health Organization learned a lot of important lessons from waiting to declare a public health emergency of international concern for the Ebola virus. And so this happened very quickly for Zika virus, which I think is a testament to our ability to respond faster when the need arises. But so this is what we're dealing with Zika virus. So we have some kind of we definitely have the human cycle, right? So it's mosquito transmitted. It can be vertically transmitted from mother to child. It can cause a lot of birth issues here, birth defects and sort of congenital neural defects here. It's unclear whether there is cycling within domestic species. But the thing that we're worried about is spill back. So if enough humans get infected and there's enough mosquitoes biting humans, and the humans are in close contact with wildlife, and we know that the wildlife has been able to carry viruses that are really closely related to Zika, the chances of spill back happening where the virus spills over from a mosquito back into a primate host and establishes what we call an enzootic cycle in the wild, this is what's going to cause Zika virus to stay in Brazil and in other countries for the long term. Right, so if once the Zika virus spills back into an animal reservoir, it's almost impossible to get rid of it. It's gonna, the potential that we're all worried about is that it's gonna continue to spill over via these mosquitoes that you can never get rid of into humans that are always gonna be there. And a solution is not to kill the monkeys. So we're worried about this problem. So we did a similar analysis. This problem was much more challenging. So for this project, I worked with the IBM Watson Research Center. They were starting up a new social good fellowship program. And so we recruited this really great student from Minnesota. He was a PhD in statistics, and he and I and the team at IBM Watson worked together to come up with new methods to deal with some really big data challenges here. So not only do we have fewer positive Zika virus positive species to train the algorithm with, we also don't have a ton of data on the features, like the life history traits of these primates, because they're just, even though we study primates and we like them and they're very charismatic, we actually know surprisingly little about their basic biology. So we had some issues to deal with the data. We used a slightly different method. We tried to work out with this method called imputation, some of the missing data issues, and we made some predictions about where we think the primates should be that are maybe suitable hosts for Zika virus. And here's their distribution. So I want to focus the results on South America here. So if you look at Brazil, the black dots represent where human outbreaks, these are human cases. Okay, so human cases are happening at all these dots. And the outlines are just the geographic ranges of the species that the algorithm predicted to be able to carry Zika virus. So what I wanted to find, well, I don't know if wanted. So I don't know if I was happy or not, or I don't want to evaluate it. But basically, we found these results and kind of like we got really disturbed. Because what we're finding is not that Zika virus is only susceptible or only suitable in those host species that are deep in the Amazon, that are rare, that that if humans are anywhere near them, their populations are gonna join. I mean, the best case scenario would have been for Zika to not be suitable hosts. For Zika to not be suitable in these hosts. These are the most common hosts that you could imagine in South America. So has anybody been to South America or Central America? Okay, so you guys have definitely seen this guy, right? I think they're adorable, but they're also kind of annoying, right? They steal your stuff, they eat your food, they get really close to you, they have no fear. They're considered what we call human commensals. because they just are not bothered by humans being around them. And so our contact with this particular species is very high. Here's its distribution, not too far from these dots. The white-fronted capuchin is very closely related. I think that these two species, the white-fronted capuchin and the tufted capuchin, the black range is here. These two are very commonly kept as pets in Brazil. So these are the species that if you wanted to, do the touristy thing and you're in Rio and you want to see a monkey, you pay a little kid who has a monkey on a string some money and you get to pet the monkey and like cuddle the monkey. These are the monkeys. Okay, so just so you know, FYI. Venezuelan red howlers, right? So these are the guys that make that really big noise in the morning, wakes you up when you're staying in the tropics. What is this? The black howler. It's not labeled here, but that's the black howler. The black howler has a pretty big distribution down here. This one is hunted sometimes, and this one is kind of a nuisance to farmers because it likes to crop raid, and it has very close contact with livestock. So these are basically the species that if you had to like reverse rank order the primates, like these are the worst ones that you'd want to be suitable hosts for Zika virus. Not only did our algorithm identify these guys as priorities for future surveillance, but it also, because of the method that we used, it identified this guy, and this guy, these three, as also suitable for carrying additional flaviviruses, which they have not been found positive for yet. So if we're going to prioritize surveillance for these species in South America for Zika virus, we should also be looking for other flaviviruses that are transmitted by mosquitoes in these species as well. So one final thing that we did was we said, okay, well, this algorithm seems to be working. We're trusting the results, but what if we pretended that the species that are positive were actually negative? Like, how good would the algorithm do it? Like, if I took away information and said, okay, we'll just pretend that this one doesn't test positive. What risk score would you assign it? And we did that for every single one of the monkeys that were tested positive. And it did a pretty good job of rank ordering the ones that are positive as positive. But some of these red ones, actually the algorithm couldn't. Even though we know already that they're positive for Zika virus or flaviviruses, the algorithm, if you don't give it that information, it can't predict very well. And when you look at what is in common across these species that make the algorithm do the worst, is that these are the ones for which we are lacking basic biological information. And that's kind of a shame because some of these species are extremely common. I mean, the analogy would be like if we didn't understand the basic biology of the red robin, because we see it all the time. How many eggs does it lay? I don't know. Like, what does it eat? Maybe worms? I don't know. It's on the ground. It's like that level of knowledge. That's sort of where these guys are at. Like, we don't have basic information about them, which I think is incredible. So our hard limit to being able to make progress in predicting the future of infectious disease is basic science. You can't predict something from nothing. And if we continue to cut our budgets and continue to devalue basic discovery over application, we're not going to be able to get very far. We're not like investing in the goose. We're just wanting more golden eggs, right? So that's the common thread that's coming out of all of these studies. We've done this type of analysis now for tick vectors, for mosquito vectors that should be able to carry Zika virus, for primates that should be able to carry Zika virus, for bats that should carry Ebola virus, for rodents that should be able to give us zoonotic diseases. We've done this now several times, and this is always a limiting factor, is where's the basic information that we need to be able to do this prediction better? And no amount of machine learning or other fancy technology is going to make up for that gap. So I recently had a little star moment where I don't get starstruck very often except by like science celebrities. And I consider this guy a science celebrity. Has anybody read this book? Yes, it's such a good book, right? Yeah, it's so good. So he's a science writer. He wrote Song of the Dodo, Spillover. Wait, did he write Song of the Dodo? Yeah, he did. And he's working on a new book about evolutionary trees. His name's David Quammen, and he was at this meeting, the spillover meeting that was in Montana recently. It was funded by DARPA, the Defense Advanced Research Programs Agency. And a bunch of us sat around, we drank some wine, we listened to him talk about spillover. We all gave talks. We sat down in a room in Bozeman, Montana, where it's beautiful, and we didn't get to go outside at all. We were in this room from like 8 a.m. to like 11:30 p.m. We filled up pages of notes and came up with all these project ideas to try and solve this problem of like, how do we understand spillover better? How can we get better at taking the data that we have and figuring out what pieces of data that we need to allow us to make the next big jump in predicting spillover events? That's what we were doing at this meeting. And I got to meet David Quammen. He was so nice. And he told us, he gave this talk, not about, the science, because, the scientists are there. And he said, I'm here to tell you how to communicate science. And he said one point that would stick with me. He said, it's kind of doom and gloom when you talk about disease. You show lots of gross pictures. You talk about how the world is getting sicker and sicker and people are going to die, and 11,000 people died in Ebola, in the last Ebola outbreak. But you have to be positive at the end. And so I thought about that message for tonight, and I thought, I'm going to go into this room full of people and tell them about all the different ways we can die, and don't cuddle monkeys and avoid rodents and all that stuff, right? But I'm really heartened by the fact that this room is full on a Friday night, and there's kids here. My one kid is sleeping. I guess it wasn't that exciting. 4-year-olds are not able to hang yet. But I'm really heartened by the fact that even though our current administration seemed to want to devalue basic science, the bipartisan vote was, nope, we're going to keep the budget flat. And we're not going to give an inch. And it was a bipartisan support for that. So that gives me hope that we're not all going to hell in a handbasket. Thank you so much for coming. Happy to take questions.
Hi, is this working? Yes. You've just said that you can't kill all the monkeys.
No.
What good does it do, in a sense, to identify which monkey is carrying the virus? How do you contain it, and how do you keep it from transmitting into the human population?
Yeah, I think did everybody hear that question? What do we do about, once you find the monkey, what do you do next? So that's a great question. There's lots of different things that people have proposed. Some of the easy ones are like, well, why is that monkey shedding virus? Is it upset? Is it because we are taking away its fruiting trees? Is it as simple as planting more trees and not cutting the rest down? And that's a really low-cost solution. Some of the more fancy solutions have things to do like with the vaccines. So there are these new crazy vaccine technologies, including an infectious virus that delivers vaccine. The way that a virus transmits, it's transmitting vaccine from animal to animal, and that has the potential to be really useful for primates because they're incredibly social animals, and they are sort of a lot of them are fission fusion. So they come together during certain times of the year. There's lots of mating and contact going on, and then and then there's fission, and then these groups have high contact. So if you can figure out, there's been network analyses done on this, if you can figure out when this happens and who the centrally connected individuals are, you could vaccinate like 5 or 10 of those guys, and you've got the whole population. That's way cheaper than getting a vaccine that you want to develop for a human through sometimes 20 years of trials and billions of dollars before you can get one that's working and approved, and then you have to disseminate it. So if you don't have perfect dissemination, you're also going to have repeated spillovers. So there's lots of ways that we can fix this, but the problem has been that we've never really thought about what to do. Like, we don't really care where it comes from. We just want to fix it when it gets to us. Like, we're putting out the fires as they come out, not thinking about how to prevent the fires from happening in the first place.
Be
What do I think the next pandemic will be? Oh, man.
like equivalent to the 1918 pandemic.
I think it's probably going to be flu again. Yeah, 1918 flu pandemic was bad. But I think if, out of all the viruses that we know about right now, the one that most people seem to be on consensus about that has a pandemic potential is flu. Yeah.
You mentioned hunting. Is bushmeat a serious problem as a vector for disease?
It depends on where. There are, some tribes that just won't eat monkeys, and it depends on what part of the world. So I think that, you touch on a really important point that, like, we haven't really considered the social element of disease transmission really before, not very often. Some of Shannon Ledoux's work does this extraordinarily well in urban settings, like the socioeconomics of who gets infected and who doesn't, right? And in West Africa, it was burial practices. We had no idea that that was the mode of transmission. And so it had, just this sort of social component is really important to consider. Yeah.
Yes. Can you say whether this spread of increase of viruses serves any positive evolutionary function?
So I don't know if I would consider it positive, but an artifact is that there is an arms race going on, right? So something comes out that seems to be winning at the moment, a virus, and then the animal responds. It either dies out or it responds by figuring out how to evolve around, co-evolve with that virus. So there is an arms race, and it's an evolutionary long-running race. My problem with it is that sometimes we could just be considered a blip in that long race, and I don't really want that to happen. So, I guess viruses killing off vast swaths of populations could eventually, in several million years, be considered positively. I don't know. That's probably not the answer you're looking for.
Hi. What do you think about the release of genetically modified mosquitoes and other vectors like that?
Yeah.
Pros and cons of that.
Yeah, so genetically modified mosquitoes, I don't know if you guys are all aware of this technology, but they're trying to develop these mosquitoes that when you release them, after one generation, the eggs are not viable, and so over time, the population starts to dwindle naturally because of this first pulse of mosquitoes. That's sort of the theoretical concept. It's controversial. Yeah, so the controversial part in the near term is how expensive it is. So the amount of mosquitoes that you would actually need to breed and hatch and then take and disseminate. It's like you spend all this money and you're like, poof. It's like a drop in the ocean, right? Like mosquitoes, and even if you test it on an island, it's like you release, however many thousands, tens of thousands of mosquitoes, and it's like, well, did that work? I don't know. And also, there's some of the technology is not. It's hard to know. You release some. It's like biocontrol, right? Anytime you have a biocontrol, there's this element of unpredictability, and you are losing control at some level, because once you release something that's alive in the wild, it's like, well, what is that gene going to do? Is it going to propagate forward? Like, you just don't know. So I have mixed feelings about it. I think Shannon is probably the person that you want to talk to about this. She's an expert on mosquito biology. I'm redirecting. That was a good question.
When you said that in your first study you controlled for like GDP output and like internet connectivity, like how did you do that?
Oh, I didn't do that. Kate Smith, smarter lady than I, did that. Let me think about this. So I'm not sure how exactly. I'm thinking that it's probably a regression style analysis, and they may have added it in as a factor. But I'm not. I can give you the citation if you want to look into the specifics. But yeah, I don't know exactly like quantitative, like mechanistically how they controlled for it.
Yeah,
They took it into account, yeah. But I think that the fact that they took it into account was novel. Like a lot of people hadn't figured out a good way to do that, and I think they were considered to be the best at having addressed it in the best way that we could address it at the time. So, yeah.
Okay. What is it in the nature of the virus that makes it do what it does, that it has an disadvantageous effect on us? Does it like to eat certain things like sugar, or what is in the nature of the virus that makes it,
Well, the virus takes over cells. It gets into a cell, takes over its machinery, and then just replicates itself, and then until it lyses the cell, right? So. What advantage does it have for the virus to do that? Why does it like to do that? It's just a fitness. It's a fitness argument. So, there's something inside of all organisms that make it want to reproduce. Like that's the Selfish Gene by Richard Dawkins is an excellent book that describes this. But it's this drive to reproduce, to increase your fitness, to make as many copies of yourself as possible, to maximize your population size. That's what drives the viruses to replicate, to maximize its replication rate, to get out of the host as many copies of itself as it can. There is a balance, though, because if you kill your host too quickly and it's not able to transmit very well because you've incapacitated it so quickly, that's not advantageous to you. So you want to kind of reach a balance where I'm going to make you sick enough to where you're still walking around and you're like, I can make it to work. I can make it to work. And you get to work and you're this perfect little, it's like this diaspora of virus particles. So there's a little bit of a balance that needs to be had there, but essentially it's a fitness argument. Yeah.
So for the machine, I'm over here, sorry.
Okay.
For the machine learning algorithm, you entered a long list of attributes for different species, and then those 5 key monkeys popped out as the most likely vectors, right? Do you know what the key attributes of those species were? Or?
Yeah, so for the primate analysis, the attributes were not so compelling. Like there were things like geographic range area, like how widespread they are. For some of the other analyses, the results were much more compelling. So they told a clear ecological story. So we did this analysis for rodents, and we got the outputs like, these are the rodents that should be able to carry zoonoses, and then here are the features that predict the ones that should carry them. And when we look at the features, it's like, oh, this makes so much sense. It's the ones that reproduce fast, that sort of live fast and die young strategy. They have lots of babies starting early. They don't gestate very long. They start reproducing at a very young age. They die early. And these things map onto their sort of immune strategy, right? If you are focusing all of your resources into having more babies, you have to make up for that cost somewhere, and your immune system is very expensive. So what these animals often, you see the trade-off is high fitness output, really general, basic immune profile, like immune strategy. So it's like sort of like this, I'm just going to tolerate infection and pump out as many babies as possible because I'm going to die young anyway. So they just kind of go out producing babies, and so it's almost like they can tolerate these infections, but when those infections get into us, if we don't have that strategy, it's really detrimental. So for the rodents, it was compelling. For the primates and Zika, I didn't get a clear signal from that. It was more of like a prediction exercise to help people on the ground who are doing the surveillance. It depends on the group.
This is some fantastic work you guys do. What safety precautions do you and your colleagues take when you're boots on the ground out in the Congo Basin capturing specimens to study? Because the fear factor, obviously, when it hits close to home, like when Ebola landed here, drives people crazy, even the emergency response folks. So we're curious what you do.
I sit in front of a computer, so I'm pretty safe. That is the God honest truth. I've spent a lot of time traipsing around in the rainforest back when I was naive to yellow fever and the fact that I shouldn't be in that jungle because it's a hotspot for dengue and all this stuff. But these guys that I know that work on filoviruses in Ghana and stuff, they go into these huge caves. I have a picture of one, actually. Can you see this cave? So these guys are on full-on PPE, personal protective equipment, with a respirator, full suits. This is like tropical Ghana. It's hot. And these are all bats. Yeah, it's really cool, huh? And sometimes it's like mixed species, like they kind of usually, I don't know if that's common. In bats it's common, but it's not common in other animals. They don't like sort of intermingle among different species in big groups like this. But yeah, so this is sort of the precaution that people take when we're in the field studying stuff that is zoonotic. Of course, you've all seen pictures of what the healthcare workers are wearing when they're out in the field. But for Ebola, it's like wash your hands. It's actually really some of the basic sort of medieval techniques. They still work. Washing your hands, quarantining yourself. Yes.
It is well known that viruses live on the genome of almost every species known. Is anybody looking at the genome of some of the targeted organisms that are thought to carry the various viruses to see if they're there? This is very well known right down to bacteria, where where the virus can be inherited through generations and suddenly becomes active and kills the bacteria releasing itself. So are we looking at the animal genomes in areas where we think these are giving rise to the various deadly viruses?
Yeah, that's a great question. So what you're describing is sort of this big push right now in basic science to understand genotype to phenotype mapping. So the idea is to take the genetic sequences of these animals and figure out how they map onto what we actually see. So do we want to know something about its productivity, if it's a plant organism? Do we want to know something about how many diseases it's carrying, if it's some kind of a rodent, and we know that it's carried other things? That genotype to phenotype mapping is a hot area of research right now. So it depends on genomics, which is high throughput sequencing, basically taking the genetic material that you find in a given organism or in a mosquito or in the environment, sequencing everything, and then putting the pieces of the sequences back together to be able to separate viruses from bacteria, from the host, from the plant that it ate, from the decomposing matter on the ground that it came into contact with. I mean, the genetic material is there's so much of it everywhere that the signal to noise ratio is really, really low. But the technology is getting better, and there are a lot of people who are doing tons of viral genomics right now. It's a very exciting field. It's very fast moving, but yes, we're working on it.
So will you join me in thanking Barbara again, and thank you for coming.